Okay, good afternoon. Just want to say a special hi to the families that might still be watching. So what I'd like to do is just kind of give an update on our contribution to the team. That's the development of antibodies and nanobodies, recognizing PPP2R5D. Our animal of choice is the alpaca because it produces an exceptionally unique antibody.
Can you get the microphone?
Up. All right, thanks. I thought I'd start out with just a short discussion about the difference between antibodies and nanobodies. I know the research team probably is well aware of this, but the more projects that I do, I'm noticing that there are a lot of colleagues, including some bright cell biologists and even some immunologists that don't really know the difference, are not fully comprehending the difference between antibodies and nanobodies. So in our case, when we put in a antigen or a target protein or peptide into our animal, it naturally produces antibodies to that target. These antibodies can be found in the plasma and then subsequently isolated and purified. And they, by nature, are polyclonal antibodies. They recognize multiple epitopes within that target or antigen. All right? In the case of alpacas, there are both conventional antibodies consisting of a heavy chain and a light chain, as well as heavy chain only antibodies. For nanobodies, we actually derive those from the peripheral blood mononuclear cells, the B cells. We use a series of molecular biology strategy, and I'll go through that briefly, to generate a fragment, a cDNA sequence encoding the minimal antigen recognition domain. So this is actually a sequence that we are pulling out from these PBMCs. And given the wide diversity of these B cells, they're monospecific, they recognize a specific target, but given the diversity of the B cells, we can actually generate a large number of monospecific nanobodies for every given target. All right, so just kind of a complicated slide. This is kind of a status of our antibodies and nanobodies. I think it might be useful for some of the team. Take a look at it. We have purified a number of polyclonal antibodies to various peptides, including the number of the variants. In addition, we have some phosphospecific antibodies that have cleaned up nicely and are very clean by a phospholipid analysis. Most recently, we made a nanobody or an antibody, polyclonal antibody against the extreme N-terminus. That looks beautiful by dot blot analysis. We have not yet tested whether it recognizes the full-length protein, but hopefully my goal when I get back, I heard it loud and clear today that I need to get these antibodies out to the rest of the team, and I will do that. We have made libraries, nanobody libraries for all these from animals, the PBMCs from these animals that were introduced that had these antigens injected into them. But what I'd like to spend the rest of the time on is just telling you a little bit of some of our nanobody progress here. And the way we go about making the nanobodies, we first isolate the RNA from the PBMCs, and then do an RT-PCR step to isolate that sequence encoding the minimal antigen recognition domain. And then we use phage display, and that's a way of screening a large number of clones for their ability to recognize the antigen. Basically, we have this large library of nanobody displaying phage. Then we pan for the phage that recognize that antigen. So we put that antigen down on a well, and then we use our mixture of phage to pull out phage, or nanobody displaying phage that specifically recognize that antigen. We can elute off that rescue the phage by putting them into bacterial cells, and then we subsequently isolate the individual clones and then induce their expression, because they're now in bacteria. We can induce the expression of this antibody or antibody fragment. We prepare the extracts, then we can screen for target selectivity. And shown here is one such example of recent data that we've been able to obtain. And this is antigen from Shang Guo and Yagna's lab. It is GST tagged 2R5D encompassing amino acid residues 512 to 602. So that's kind of the N or C terminal domain. We actually coated the plate with that, and we were able to detect a fairly large number. We picked 96 clones just randomly. A1 and A2 are always controlled antibodies and irrelevant antibodies, so they'll serve as our control. So we were able to get some positive signals for all those, but the question comes up is, are those antibodies recognizing this fragment of 2R5D, or are they recognizing GST? GST is a tag that's on there. So to address that, we actually coated a plate with GST, and we were able to see some that recognize the fusion protein as well as GST alone. So those are likely GST recognizing nanobodies. And the others could very well be nanobodies that recognize 512 to 602. And we need to follow up on this, hopefully get some more purified prep that we can further test this. Another example, a little bit larger fragment, this is 2R5D encompassing residues 98 to 513. This was largely untagged, but there was a fair amount of GST tagged protein in there. Forget how much exactly, 10, 15% of that was in that preparation. That was put into animals. We were able to get a number of clones recognizing that antigen. And we also tested for GST. And again, we could see some difference between the antibodies recognizing the GST fusion protein versus GST alone, which could again be nanobodies recognizing that epitope or that fragment of 2R5D. So just in the last 5 minutes or so, I'd like to tell you about another project that's not directly related to 2R5D yet. It could be here in the future, but I think it's very relevant to the idea of trying to make nanobodies against these individual variants. And this goes back to a project that's probably dogged me for the last decade or so, and it has to do with tyrosine phosphorylation of PP2A. At our first meeting in Scottsdale, I think I showed, actually, let's start out. First meeting in Scottsdale, I talked about the tyrosine phosphorylation of PP2A and data demonstrating that it gets phosphorylated on two sites, Y127 and Y284. And one of the things that we were lacking at the time was antibodies. So we went ahead and tried to make an antibody. This is a polyclonal antibody against the phospho-Y284 peptide. We put that into alpacas, isolated the plasma, purified the antibodies, polyclonal antibodies, from the animal. And the way we did that, take the plasma, pass it over to the non-phospholipeptide column first, take the not bound from that column, pass it over to phospholipeptide column, elute those off, and then test for their specificity. This is simply dot blot analysis. We're able to purify antibodies that specifically recognize the phospholipeptide that failed to detect the non-phospholipeptide. We also had antibodies made in rabbits for the other site, phospho-Y127. Here's just a control antibody recognizing BSA, phosphorylated on tyrosine residues. Wherever you have tyrosine phosphorylated BSA, it's detected with that antibody. Okay, so the next question, actually we use that antibody, one more piece of data related to these antibodies. We use, and we're able to detect where we introduced the HA-tagged PP2A catalytic subunit into cells, and we could detect a phospho signal when we co-express constitutively active SARC. If we mutated the Y284 site to a phenylalanine where it no longer can get phosphorylated, we lose recognition of that site, indicating that it is indeed specific for that site. So these polyclonal, affinity purified polyclonal antibodies have been extremely useful for our research purposes. But the question that came up and I kept asking myself is, can we make nanobodies against that epitope? And this was something that, according to literature, sounded pretty challenging. People have said it's usually it goes back to one report saying that it's very difficult to make nanobodies against linear epitopes, or small epitopes. But if you don't try, you can't succeed. So we actually did go ahead and try to screen for nanobodies recognizing this phosphoepitope. We took the library and generated the phage, did the phage display, and then actually screened. What we put down on the plate was the phosphorylated Y284 peptide conjugated to BSA. Okay, and that's pretty clean prep. Just BSA with this phospho Y284 peptide covalently attached to it. And we were able to see, again, the controls, A1 and A2. There's a large number of positives that lit up. The question that then comes up is, how do we know that it's phosphospecific? It could be recognizing the non-phosphorylated peptide. So we put down the non-phosphorylated peptide, and we were able to see a very good difference between these clones. These are the same clones probed or using a plate, a phospho Y284 peptide plate versus a non-phosphorylated peptide coat. And you can see a large number of what appear to be phosphospecific nanobodies. So the reason I'm really excited about this is because that kind of suggests that, hey, all those variant peptides that we put into the animals, it might be possible to generate nanobodies or monospecific antibodies against those variants, which could prove to be very useful in further studying it and possibly even modulating the activity of the 2R5D holoenzyme. I think that's all I have, just to acknowledge the people. I got 3 people that are kind of been working on this: Sarah Kahn, Anthony Jordan, and Matt Duvernay. And then, of course, Jordan's Garden Angels for the support on this. Happy to take any questions. Gotta always end with a picture of the 4-legged creatures there.
So Brian, I guess the one question I have is, you heard a lot of folks, do you kind of know who you need to get to? Just want to make sure we're helpful with that.
I do. First thing, I'm getting back next week, and that's on the top of my list. What I'd ask everybody to do is just send me kind of the wish list. Go back to that long, thing. Anything that I have there, the polyclonal, we have plenty. We can send you a good mil of that stuff, along with the secondaries. We got to do a little bit more work on these nanobody clones to see if they're truly real, but then we could use help in further testing them. Those might even be screened or tested to see if they bind to the holoenzyme, and possibly might even be able to stabilize the structure as you were